Okay, I'm going to try something a little bit different when we go over these free response questions for chapters 9 and 10. I've kind of cut and pasted part of the rubric and what it's requiring into these answers here, so we'll see if this will help at all to just kind of see all the little pieces that the rubric is requiring. This one was talking about a manufacturer of toxic pesticide granules, and they're planning to use this dye to color their pesticides so that the birds would avoid eating it. A series of experiments will be designed to find color or patterns that 3 bird species, blackbirds, starlings, and geese, will avoid eating. Representative samples of birds will be captured to use in the experiment, and the response variable will be the amount of time that a hungry bird will avoid eating food of a particular color or pattern. For part A, it says, previous research has shown that male birds do not avoid solid colors. However, it is possible that males might avoid colors displayed in a pattern, such as stripes. In an effort to prevent males from eating the pesticide, the following 2 treatments are applied to the pesticide granules. Treatment 1. Red background with narrow blue stripes. Treatment 2, blue background with narrow red stripes. Okay, to increase the power of detecting a difference in the 2 treatments in the analysis of the experiment, the researcher decided to block on the 3 species of birds, blackbird, starling, and geese. Assuming that there are 100 birds of each of the 3 species, explain how you would assign birds to treatments in such a block design. Okay, the key here is we are trying to increase power. This is something we learned about in chapter 9. Okay, so here's what the rubric had to say about that. Okay, for this, it said if you wanted to get an E on this, it says that the student has to describe a method for how you would randomly assign the 2 treatments to the birds within each of the 3 blocks, and you have to describe that method with sufficient detail so that a knowledgeable statistics user could replicate the method. So if you just simply said, oh, I would randomly assign half the blackbirds to treatment 1 and then the other half to treatment 2, and then I would do the same thing for the starlings and the same thing for the geese. That is not using sufficient detail so that someone could really replicate your method. We need something like this. Tag each of the 100 birds within each block with a number from 1 to 100, then use a random number generator to generate 50 numbers between 1 and 100, skipping repeats and numbers outside the interval. Those corresponding birds will receive treatment number 1. The remaining 50 birds will receive treatment 2. Repeat this process for each block, blackbirds, starlings, and geese. Okay, that would get you an E if you have that kind of detail. Okay, you have to remember on the AP test, communication is key. Okay. Part B here, it says, other than blocking, what could the research do to increase the power of detecting a difference in the 2 treatments in the analysis of the experiment? Explain how your approach would increase the power. Okay, see how it has this extra part? So you're going to say what you would do, and then you're going to explain how that would increase the power. Okay, here's one option to get an E. Okay, you could say the researcher could increase the sample size. Okay, if you stopped there, you are only going to get a P on this because you're not linking how that's increasing power. Okay, you could say something like, when we increase the sample size, we are decreasing the variability, we're knowing more about the population. In turn, that increases the likelihood, remember this is what power is, correctly rejecting a false null hypothesis. The other option that would give you an E would be to say that to increase power, you could increase the alpha level. Okay, and again, you have to link this. By increasing the alpha level, the researcher could make it easier to reject a false null hypothesis. Okay, when you reject a false null hypothesis, that's what power is. Okay, that increases the power of the test. Okay, our next one here is talking about a recent report. It stated that less than 35% of the adult residents in a certain city would be able to pass a physical fitness test. Consequently, the city's recreation department is trying to convince the city council to fund more physical fitness programs. The council is facing budget constraints, and it's skeptical of this report, so they're only going to fund a physical fitness program if the recreation department can provide convincing evidence that this report is true. So the Recreation Department plans to collect data from a sample of 185 adult residents in the city, and a test of significance will be conducted at the 0.05 level. Okay, so here's what they're after. P is the proportion of adult residents in the city who are able to pass the physical fitness test. So our null hypothesis is that 35% can pass it. The alternative is that less than 35% can pass. Okay, what part A is asking, it wants us to describe what a type 2 error would be in the context of this study, and then also describe a consequence of making a type 2 error. Okay, I went ahead and made a note. A type 2 error is when your null hypothesis was actually false, but we failed to reject that. Okay, so to get an E, you have to describe the error. It has to be in context, okay? So we could say a type 2 error is when we say that we do not have enough evidence to say that the proportion of adults in the city are able to pass the test is less than 35%. So keep in mind, we're failing to reject this. That means our conclusion is we don't have evidence that less than 35% could pass it. But when in reality, the truth is only less than 35% can pass it. Okay, that's describing a type 2 error. A consequence for this error is that the council would choose not to fund the fitness program, thinking that we at least had 35% who could pass the test, when in all reality, the residents really are in need of the fitness program. Okay, right, so that's part A. Part B, it says the recreation department recruits 185 adult residents who volunteer to take the physical fitness test. They do the significance test. They end up getting a p-value of 0.97. The question is, what would the p-value of 0.97 lead you to conclude? Okay, keep in mind, up above in the problem, they're telling us that the alpha level is 0.05. We would look up here and make note of what our null and our alternatives are. Okay, and this is just our standard, the way we end all significance tests. You'll notice that this is asking you, look, to get an E, you have to link the p-value and say that that is greater than the alpha level, which is what we always do. We would say since the p-value of 0.97 is greater than the alpha level of 0.05, we would say we fail to reject the null hypothesis. Okay, then for these other two parts to get an E, you have to make the correct conclusion that describes you don't have enough evidence for the alternative, which again is something we always do. We don't have convincing evidence that the true proportion of adult residents who could pass that test is less than 0.35. And you'll notice the second part here is you have to have context, and that's where we're commenting the true proportion of adult residents who can pass the test. Okay, that part was pretty easy, hopefully, for you. Okay, part C says describe the primary flaw in the study described in part B and explain why it is a concern. Okay, well, the issue here is, first of all, you have to say that the sample was not random. They were a bunch of volunteers. Okay, so that's the problem. Okay, but then you have to explain what's the big deal with that? Why is that a concern? Okay. And the issue is you have to state how you think those volunteers would in some way be different with regard to physical fitness. Okay, so maybe saying something like, well, it's likely that those who volunteered to take the physical fitness test, they're probably more physically fit since they might be excited to take the test. Okay, and then look at part 3 here to this. You have to state that your sample would overestimate your true proportion, okay, or that it would just not be representative of the population. This would cause the results to overestimate the true proportion of adult residents who are able to pass the test, because we've got a bunch of physically fit people who have volunteered, essentially. Okay. All right, on this one, this is talking about a researcher. He believes that treating seeds with certain additives before planting can enhance the growth of plants. An experiment to investigate this is conducted in a greenhouse. From a large number of Roma tomato seeds, 24 seeds are randomly chosen and 2 are assigned to each of 12 containers. One of the 2 seeds is randomly selected and treated with the additive. The other serves as a control. Both seeds are then planted in the same container. The growth in centimeters of each of the 24 plants is measured after 30 days. These data were used to generate the partial computer output below. Graphical displays, this is important, graphical displays indicate that the assumption of normality is not unreasonable. What they're telling you here is they're giving you the check off that this meets the normal condition. Now, if you look closely, though, at the information in this computer output, they've got the mean, standard deviation for the control group, same thing for the treatment group. But look at this row here. This is the difference in control and treatment groups. Now, look at what the question's asking. The question says, construct a confidence interval for what? For the average difference in the growth in centimeters of the plants from the untreated and treated seeds. Okay, so to get an E on this, you would have to identify the appropriate confidence interval by name or by formula. We've always done by name. This would be a one sample T interval. And the reason it's one sample is because we've compiled the differences in the two treatment groups and we're looking for the average difference. Okay, that is not the same thing as the difference between the means. Okay, we're looking for the average difference. That's just the data in the difference row. We're using only that row. It is a one sample T interval. Okay, then we would have to check the appropriate conditions. This is basically telling you here that you can assume that it's normal because we were given that in the problem. We also would want to state that the seeds were randomly assigned to treatment groups. Okay, that meets the random condition. And remember, you do not have to do the 10% condition if we're talking about experiments. Okay, the do step, you're welcome to just run the one sample T interval on the calculator, or you could use the numbers plugged into this formula. Here's your interval, and then of course we always interpret. I'm going to draw your attention to what I have in this red box though. You cannot say we're 95% confident that the difference is, you have to say we're 95% confident that the average difference in the growth of untreated and treated seeds is captured by that interval. Okay. All right, part B, it says, based on the confidence interval in part A, is there sufficient evidence to conclude that there is a significant mean difference in the growth of the plants from untreated seeds and the plants from treated seeds? Okay, this is talking about statistically significant, which is usually a phrase that we reserve when we do SIG tests, not necessarily confidence intervals. Okay, while it's not required that you state hypotheses, I think it could be helpful for you to think through what those hypotheses would look like, because what this is basically asking is, for our null hypothesis, if we were to do a hypothesis test for this, we would say that there is no difference in the the mean difference here. Okay, that's just 0, versus there would be a difference for the alternative. Okay, now if you look at what our confidence interval was, notice our confidence interval does not include 0. Okay, so we could say something like, since the 95% confidence interval does not include 0 as a plausible value, then we would have rejected this null hypothesis. Anytime you reject a null hypothesis, that's when you say the results are statistically significant, that there is a mean difference. Okay, this last frappy kind of bothered me a little bit. It was basically talking about patients who have heart attack symptoms and how they get to the emergency room. Okay, whether they get there by ambulance or whether they self-transport themselves here. Okay, what I didn't like about this is it mentioned that there was a random sample, but then you can see down here they split them into these two groups. This was not an experiment, and I think this was a little tricky to figure out what inference procedure you were supposed to use. They were wanting you to use a 2-sample T interval because they took that group of 150 and they separated them based on how they got to the hospital. Okay, you can see this here on the rubric, that it's these checks of naming it correctly, central limit theorem, both groups were more than 30. I just ran it on the calculator, got this for the degrees of freedom. And again, just drawing your attention to the fact you want to make sure when you interpret this, that this is capturing the true difference in those mean weight times there. Okay, and then down here at the bottom, really similar to the other problem we just looked at, this again is asking us, based on the confidence interval, do you think the difference in the mean weight times is statistically significant? Again, if you think back to what the null hypothesis would look like, that means we would be looking for whether 0 was actually included in our confidence interval. You can see that our confidence interval right here did not include 0. That means we would have rejected the null hypothesis. Okay, therefore, these results would be statistically significant at the 0.01 alpha level. That's because it was a 99% confidence interval.